All right, welcome to our third lesson in our genetics unit. We're going to talk about the regulation of gene expression, when you turn genes on, how you turn genes off, and how you do that differently in different organisms. So I have an analogy to start us out. So let's say that I sat you down for dinner and you were eating a plate of spaghetti. What utensil would you use for this? Well, hopefully you would choose to use a fork as pictured here. Now let's say I changed that and instead of spaghetti, I gave you a bowl of soup. Would you be able to use the fork to eat that soup? Unless it's super chunky and has very little liquid, you would not. You would need a different tool for that. In this case, you would need a spoon. Similarly, if you're a bacterial cell or any type of cell, you're going to want to have the appropriate tools to use when you are digesting different food sources. So here what we see is a bacterial cell that's surrounded by glucoses, represented by those hexagons. When it is digesting those glucoses, it will need to make a specific set of enzymes. So here, this bacterial cell will make enzymes to digest glucose. Would it make sense for it to have a whole bunch of other tools there? Would it make sense for it to have enzymes to digest every single type of sugar? Not really. That would be like sitting down to a plate of spaghetti and having a fork, a knife, a spoon, and every single other utensil type you could imagine. While you go to a restaurant and they give you all the utensils, if you're eating at home, you're not going to take out every single utensil in your kitchen to eat a plate of spaghetti. You're going to take out the one tool that you need. You're only going to take out the other tools like a spoon or a knife if you need them to eat your food. It's otherwise kind of wasteful. You have to do a lot more dishes. And if you're anything like me, that's, your waking nightmare. On the other hand, if you're a bacterial cell, making a whole bunch of unneeded enzymes would not only be useless, it could be lethal, because for every enzyme you have to make, you have to spend energy to make it. And if you can't get that energy via food, then you could spend more energy than you take in, and that would result in death. So a bacterial cell, like a human, would need to use specific tools based on the food source. So a lot of bacteria, when they're surrounded by a source of glucose, are fine. They make a specific set of enzymes. But let's say we change it up, and instead of having glucose, they have a different sugar. If they have a different sugar, they're going to need a different set of enzymes. So in this case, they might bring out a separate tool. In the case of a bacterial cell, what that means is they might make different enzymes. So if you think about the genome, you can think of it almost like a giant toolbox. It has all of the proteins that you might need for any of the circumstances that you would come across. However, it doesn't make sense to make every single one of those all the time because you would be wasting a lot of things. Not only would you be wasting energy, you would also be wasting resources because to make each of those proteins, each of those enzymes, you need amino acids. You're taking in most of those from your environment, or you might be making some, and it would be counterproductive to waste those. Instead, if you have a different organism who is able to use all of their resources more efficiently, only make a particular set of enzymes when they need it, they're going to have an evolutionary advantage because they're going to be able to do more with the same amount of energy. And so we can link all of this back to evolution. If you are more thrifty with the enzymes you make, you only make the ones you need, you only use the parts of your genome that you need at a particular time, you have an evolutionary advantage. You are fitter. You are able to survive and pass on your genes using the same set of resources or acquire resources more efficiently, and you're able to pass on your genes. So with that in mind, let's talk about how organisms regulate the expression of their genes to make sure that they can have the appropriate set of genes active for the appropriate environmental circumstances. This process is, as everything else we're going to talk about, different in prokaryotes and eukaryotes. It's going to be much simpler in prokaryotes, so we're going to learn about the basic unit of the regulation of gene expression, the operon. So an operon is kind of a set of genes that are controlled by the same kind of on-off switch. So you have some sort of regulatory DNA. Usually it's going to produce a gene that's either going to turn on or turn off an operon. It's going to have a promoter. A promoter is going to be an area that kind of helps binding of RNA polymerase. A lot of times it's where the RNA polymerase is going to bind, and I tend to think of it as an on-off switch. You also have something called an operator. An operator is where these regulatory genes will bind, so that will determine whether or not these genes will be expressed. And then you have several related genes. So we're going to talk very often about the lac operon, which is involved in digesting lactose. So lactose is different sugar from glucose, and you need a different set of enzymes to digest it. So bacteria will only make lactose digesting enzymes, which are all found in this lac operon, when they don't have enough of a different source of sugar, so they don't have enough glucose, and there's lactose present. So most of the time, this gene is turned off. However, if you change the food source of a bacterial cell, it'll turn on. So let's look at that in a little more detail in a moment. Before we get to that, let's talk about a few terms. So an activator is something that is going to turn on a gene, make it more likely that it will be expressed. A repressor is something that is going to turn off a gene or decrease the chance that will be expressed. Activators and repressors can have other molecules that can help their function. So an activator could link with an inducer molecule, which would then turn on that activator and allow it to turn on expression of genes. And repressors could be either activated or they could be activated by a co-repressor, which would help them block expression of a particular gene. So we have two different types of operons that are found in prokaryotic cells. We have ones that are inducible. They are normally turned off, but they can be induced or encouraged to turn on. So they are usually blocked. They are usually repressed. And you'll need to remove a repressor for them to become active. And the classic example of this is the lac operon. So the lac operon has 3 genes that are linked. And expression of these 3 genes is either turned on together or turned off together. It makes sense to have the same kind of on-off switch for these genes because all 3 of them are needed for lactose digestion and only needed for lactose digestion. So what you have, we're going to focus on the promoter and the operator here. The promoter is where RNA polymerase can bind. So RNA polymerase would have to bind in order to move along and transcribe RNA. Without that RNA polymerase binding, you have no mRNA made. Without mRNA, you're not going to make any proteins. However, even though an RNA polymerase can bind to the promoter, it has to be able to pass this region called the operator. If it is blocked by the operator, say if there is a large repressor molecule bound here, then RNA polymerase is not going to be able to pass by, and it's not going to be able to transcribe the DNA for LAC-Z, LAC-Y, or the LAC-A enzymes. What will then happen, though, is if you have a lactose molecule, if you have lactose present in the cell, it can bind to that receptor and block its activity. That repressor. When it binds to a repressor, the repressor will no longer be bound to the operator, and the RNA polymerase is free to start making copies of the enzymes that are needed to digest lactose. So with the lactose present, you turn off the repressor, which means you're turning off the thing that's blocking you from being able to make copies of this operon. So that's how you turn on. I know it's kind of complicated where you have repression and then you're repressing the repression so things are turned on. And it may seem like it's really, is it why couldn't you just have an activator, something that turns on this gene when it's needed? Keep in mind, not everything is 100% efficient because all of this stuff, none of it was consciously designed. It's all kind of evolved. So sometimes you might have some minor inefficiencies or things that don't make 100% sense to us. The other type of operon that you will find in bacteria is a repressible operon. So this is something that's turned on all the time, but when you no longer need it, you can turn it off. It's about being efficient with your energy. So the classic example of this is the trip operon. The trip operon is going to have enzymes that are needed to make an amino acid called tryptophan. And tryptophan is something you do need constantly for building proteins. However, let's say you have a ton of tryptophan built up. If you have a lot of tryptophan, you don't want to keep making extra, because if you make too much of it, now you've got extra tryptophan sitting around. You've used up your energy to make it, and it's serving no purpose because you don't need it. So what can happen is, in this case, this this operon is normally turned on. However, you can have something like a co-repressor. So for example, tryptophan can bind to a repressor, and when that happens, it can block the transcription of these tryptophan producing genes. So then what can happen is, when tryptophan is going to be present in less concentrations, lower concentrations, because you've not been making it, you no longer have tryptophan, you no longer have an active repressor, and so now RNA polymerase is free to bind here and start making RNA copies. And so you can turn off that gene when you need to, or those sets of genes. I hope that wasn't too super complicated for you, because it's about to get a lot more complicated. Everything is more complex in eukaryotes, including regulating gene expression. So instead of having like a single promoter and maybe a single repressor with a co-repressor, or a single activator and an inducer molecule, you now have what are called series of transcription factors. So a transcription factor is a protein that will help regulate transcription. Usually when you have particular transcription factors bound to a segment of DNA, they will increase the affinity of that region for RNA polymerase, meaning they'll make it almost more sticky for RNA polymerase to bind. RNA polymerase can then come and bind to the DNA sequence, as you see right here, and can then transcribe the DNA. This gets more complicated because it's not necessarily a single transcription factor will start making you produce a ton of the mRNA. You might need a specific combination of transcription factors. There's no specific combination of transcription factors that you will have to memorize for the test, so at least kind of calm down with that. So, as we talked about with prokaryotes, activators are going to increase the likelihood that a gene will be transcribed. Repressors are going to decrease the likelihood that it's transcribed. You can get combinations of activators and repressors, and it just kind of depends on which one you have more of. If you've got more activators, then it'll make it more likely that RNA polymerase will bind. If you've got more repressors, might make it less likely. Another thing that kind of complicates our understanding of regulation of gene expression in eukaryotes is the fact that you can have very distant elements called enhancers, regions of DNA that are far potentially from the gene that you're looking at, and these enhancers could be required when they're bound to certain transcription factors, could be required to help turn on the production of a particular gene. This is kind of complicated because since the enhancers or other control elements don't have to be right next to a gene, it can be kind of hard to find them. So when you have people who are still looking at the human genome and trying to figure out what each sequence of DNA is doing, this is one of the things that they're discovering. Oh, this region is an enhancer for this particular gene. So it can take a lot of time to try and figure this out. The human genome's been decoded for the entirety of your lifetime, and we still don't know 100% what all of the DNA is doing. So, possible career options for you guys. So let's talk about some of the sequences that you find in DNA that are going to make it more or less likely that a particular gene will be transcribed. So you have these regulatory areas that are going to be binding sites for transcription factors, and depending on which transcription factors they bind, whether those act as activators or repressors, like I told you, that's going to determine whether or not you have a gene transcribed. So what we have at the top of this diagram right here is we have a number of the different elements that can affect whether or not a protein is transcribed. So you can have upstream enhancers or silencers, which are located before the gene. You can have downstream elements that can enhance or silence a gene. You can have proximal ones, which are located very close by. They can be located next to the promoter, within the promoter, and then you'll also have some terminal sequences that will indicate, hey, end transcription, we're done here, stop making mRNA. And then, of course, you have modifications that happen after transcriptions. That's what we were talking about with the pre-mRNA processing. And then you'll have ultimately your mature mRNA being used to translate and make a protein. So, like I said, fairly complex. The region of the DNA where you'll usually have RNA polymerase bind is called the promoter. So if you have elements bound to the promoter that increase the affinity or the binding of RNA polymerase, they're going to make it more likely that that gene is going to be transcribed. A common element of this is the Tata box. Yes, I know it sounds bad, but it's named that because it's a repeating series of thymines and adenines. So it's literally just a sequence of TA, 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 which is an area where it's very common for transcription factors to bind. Then you also have sites located at the end of these coding regions, which will indicate transcription is over, and they will be areas where you have very low affinity for RNA polymerase. So let's say you have an RNA polymerase kind of coming along and reading, and it is moving along this DNA segment. At this point, the termination site, that RNA polymerase will no longer bind well to the DNA. It'll come apart from the DNA, and it will stop making an mRNA. So that's why when you're making mRNA, you don't just make a copy of an entire chromosome. There's a region that tells you, hey, stop, we're done. So enhancers are areas that can be either close to or far away from a gene, and they'll usually have some sort of transcription factor that binds, and sometimes they have to actually, like the segment of DNA has to be bent, so that way this enhancer with its associated transcription factors can then bind to an area and increase the chance that an RNA polymerase will bind. It's all, like I said, fairly complex. You could spend your lifetime researching the interactions of all these transcription factors, and some people do. Any regulatory gene is something that produces a protein that will either activate or repress another gene. You can also have RNA sequences that are designed to help regulate whether a protein is translated or not. We're going to talk about a very fairly well-researched example of this, microRNAs, on the next slide. There are a number of different ways that you can, basically any step in the process of creating a protein, you have methods that the cell has to try and regulate whether or not that translation or protein synthesis is going to happen, and also ways to slow it down or speed it up. Your cells are very good at controlling with a very, very kind of fine precision when, where, and how much of a protein is produced. And so all of these things are factors that are involved. So I just wanted to talk about RNAi, or RNA interference. That is when your cells produce these microRNAs. These microRNAs will then be complementary to part of a target mRNA, and the binding of this microRNA is a signal to the cell to either stop translation or to break down this mRNA. So that's something that can be used to, let's say you've got a gene that you only want to translate for a short period of time. You could start producing an miRNA, a microRNA, very soon after you make the mRNA, and as soon as it binds, it can turn off production of that protein. miRNAs, or microRNAs, and RNA interference also have a role in immune systems because they can help target rogue RNA sequences that have been introduced into a cell through viruses. So there are a couple of different functions for these microRNAs. All right, I hope that your brain is not too fried, and our next video will be coming out soon.